Hi, thanks for your interest in my work. I'm George Ruck, working at Inrae in the ecotoxicology lab in Lyon, France, in partnership with ViewPoint Biosurveillance using their innovative surveillance tool, ToxMate. I'm here to pitch my work on bio-innovative wastewater surveillance. So the context of my work is the need for real-time effluent surveillance for wastewater management. Micropollutants and CECs in small quantities can have a detrimental impact. And on top of this, I'm interested in the transient pulse nature of their discharge. So typically, a 5-hour episode of high concentration in a micropollutant would go unnoticed. And given that there are thousands of potential candidate CECs that might be at the root of an issue like that, we believe that bio-innovative, non-targeted, effect-based methods are the way towards real-time surveillance. So we've developed this biotest using the ToxMate apparatus, which ViewPoint developed previously for real-time wastewater surveillance. This is what the ToxMate looks like. It's a 3-panel observation chamber with cameras on each panel. The chambers are either linked to a bypass of the wastewater effluent or to a water reservoir in the case of a laboratory experiment. Each panel has 16 individuals from a different species in each panel. For my work, I used the 3 species listed below. And looking at the top right, this is the minimal activity state. So the conditioning that we do in the lab upbrings organisms to be in this minimal activity state in normal conditions. And now in the bottom right, this is avoidance behavior. And this is actually the biomarker that we study to capture the micropollutant presence. So we measure the center of gravity of each organism and combine these measures to give one overall activity signal for each species. And finally, for some of our most recent results, so for Gammarus fossarum, the amphipod used in my experiments, I'm showing 7 CEC avoidance signals. Each signal comes from the 16 measured Gammarus trajectories, and what's interesting is the difference in each CEC response. So currently, work is looking to combine the 3 species responses for avoidance and investigating behavioral fingerprinting. Now, fingerprinting is the idea that the form of the response that we see is due to the chemicals present. So the 7 different responses we see are because of the differences between those 7 chemicals. And we can use, we hope to be able to use fingerprinting for contaminant identification later on. So the next step is to take this to treatment plants. We have operational ToxMates across France and Europe with alert systems in place over the last couple of years. And the long-term data that we've collected provides a very unique and valuable insight into the evolution of effluent nature. So if you'd like to find out a bit more about my work, get in touch, have a look at my poster, or get in touch through the following details. Thanks a lot for your time.